Hello, I'm Dave Bartel, and welcome to the third part in this three-part series on microRNAs. And in this third part, what I'd like to do is discuss some of the issues of deciding what's a microRNA and what isn't. So our lab and many other labs have been looking for microRNAs for over the years, and we've developed different approaches for doing that. One is to computationally look for conserved hairpins in the genomic sequences and see if those have the features of microRNAs. And the other approach is just to sequence the small RNAs that are found in different cells or tissues or species. And the real advances in this microRNA gene identification has actually come in the sequencing approach, and that has been because of high-throughput sequencing, which has just been available for the last 5 or 6 years. So this slide just shows kind of a timeline of our sequencing efforts in C. elegans. So back in 2001, we were really quite happy with what we could learn from 330 reads matching small RNAs in C. elegans. And that included 10 reads to miR1, as well as reads representing another 50 additional C. elegans microRNAs. Then, a few years later, we scaled up this approach, and then we started seeing reads matching the other strand of the hairpin, what's called the microRNA star strand, present at lower abundance, but with more sequencing, you can start to see that. And as we're starting to see these what are called microRNA star reads, we got some of the early evidence showing that the microRNAs are being processed through this duplex intermediate that I described in the earlier talk, the introduction. Later, then a few years later, the high-throughput sequencing from the 454 technology was developed and available, and we were able to increase our throughput another 100-fold, and then more recently, even higher throughput technologies become available. This is Illumina sequencing, where we have over 22 million reads from C. elegans. And other groups have now extended this even more. So at this point, we have over 1 million reads to miR1. And of course, the reason for doing this isn't to get the same microRNA again and again and again 1 million times, but it's to find microRNAs that had been missed in earlier efforts, maybe because they're only in a few cells or expressed just in very specific times in development. And so we've been doing that, and in the process of doing that, we've had to develop annotation criteria, because when you get these millions of small RNA reads, it's possible that just by chance, some of them are going to map to things that look like they could fold into hairpin precursors, and there's a danger of getting false positives. And so the criteria that we've been using to decide what's a microRNA hairpin and what isn't is to look for, first of all, multiple reads matching a predicted microRNA hairpin. Okay. And then second, we look for reads where the more abundant read has a very consistent 5' terminus. Okay? And that's important, because if you remember the way that the targets are recognized, what's important is pairing to nucleotides 2 through 7 or 8 of the microRNA. Okay? And if the 5' nucleotide is shifted 1 up or 1 down, that's going to change that seed sequence, which is going to really have a profound effect on the predicted targets. And so what we see, and what makes sense, is that the authentic microRNAs have a very consistent 5' end. And also what's really useful is identifying reads from the other arm of the duplex here from the sort of microRNA star arm. And so if you get reads from both arms of the duplex and see that they map to this hairpin with 2 nucleotide 3' overhangs, which are the sort of signature of Drosha and dicer processing, that becomes very good evidence that these are indeed Drosha and dicer substrates, and that this hairpin has entered, at least some molecules have entered the microRNA pathway. So these types of criteria then have been used by our lab and other labs to find additional microRNAs in animals and plants, and we've applied it to find additional microRNAs in C. elegans, Drosophila, mouse, Arabidopsis, moss, and identify microRNAs in lycopods, another early branching land plant, sea anemone, sponge. And so this has, I think, been very, very, very useful. And here I'll just show you an example that we got looking for additional microRNAs in mouse. So, and I'm just going to be focusing on the canonical microRNAs. There are additional microRNAs that are, again, processed sort of in a way that bypasses the Drosha processing, but what I'm going to focus on are those that appear to be canonical microRNAs, probably processed first, the primary microRNA processed by Drosha and then dicer. And what you see is that we have very high overlap with what had previously been found in mouse. There had been a lot of work, particularly by Tom Tuchel's lab in the mouse, and many microRNAs that have been identified, and also by other labs. And what we found was that there was, we found support for 377 of those microRNAs that were already in this database, which is called MIRBase here. So this is a database of microRNAs, and at that time there were 554 microRNA genes that had been annotated in MIRBase. We also found additional microRNAs that hadn't yet been annotated, so that was nice. But there were also nearly 200 annotated microRNAs that we didn't have support for. So why would this be? Well, even though we had 60 million reads from a diversity of cell types, it could be that we were just looking in the wrong cell types. And if we had looked in other cell types, we would have found some more of those. And that could have happened. Or it's possible that our annotation criteria were actually too stringent. And because many of these things that had previously been annotated, not by Tom Tuchel's lab, but by other labs, really didn't meet our criteria for annotation. But of course, it's hard to know where to set the line in these annotation efforts. And so what we decided to do is start to investigate experimentally which of these have the features of microRNAs. And the way we do this is just to overexpress them. So previously methods have been developed to overexpress microRNAs, and we could just use those methods to express them in mammalian cells, where we would place the candidate hairpin behind a strong promoter so that we would know that that hairpin is being expressed in these cells. And of course, these cells have the Drosha, they have the dicer, and so if that RNA can be recognized by the microRNA machinery, it's going to give rise to small RNAs, and then we can sequence the small RNAs. And we actually did this sort of in a medium throughput method, putting multiple candidate hairpins into the cells, and then looking all at once at the small RNA sequencing. And so here are the results of this experiment. Of course, we did a bunch of positive controls, and those are shown here on the top. And these are known mouse microRNAs, very confidently identified, highly conserved. And in this cell type, each of them was highly expressed, much more than in red, which is what was just found endogenously in the cell type. Note that this is a log scale in terms of number of reads. And so you can see here that these blue bars are much bigger than the red bars and much bigger than 0. Okay? And that's what you see for the controls. Even though these controls, many of them are very specifically expressed only in very specific cell types and not in the cell type that we were looking at, we could overexpress them in this cell type. What about the microRNAs that we didn't have support for because we didn't get any reads for them? Well, there we could not overexpress them. So they don't seem to have the features that are needed for Drosha dicer processing. Probably not microRNAs. Now you can imagine how we get some false negatives in this type of assay, but the fact that so many of the controls checked out means that we think that the vast majority of these probably are not actually microRNAs, just false positives in the database. And we got similar results for cases where we got a few reads, but they didn't match our annotation criteria. So as molecular biologists, we think we have pretty good, it's not perfect, there are annotation criteria, okay? We might miss some authentic microRNAs with this, but we think that we're getting rid of a lot of the false positives. So, and this is still evolving some, but we think we have a pretty good idea as molecular biologists in deciding what's a microRNA and what isn't when going through these many millions of small RNA sequencing reads. But really the more important question, the more interesting question is, how does the cell make this decision? How does the cell decide which of these hairpins should go into the microRNA pathway and which shouldn't? And there's a lot of work now showing that much of the genome is transcribed. There are many parts of the genome that when transcribed would fold back into these hairpin-like molecules. And how does the cell decide which of these molecules are actually primicroRNAs and which should just be ignored? And this question became even more interesting when we saw that there's very different criteria for human cells and nematode cells. So when we, again, we're using this overexpression assay where we put in human microRNAs into human cells, no problem. We often see expression of those microRNAs at different levels, but still, because we have such a dynamic range in this assay, we really see plenty of expression for most of the human microRNAs that we try. But when we tried to express these hairpins, microRNA hairpins, known microRNA hairpins from C. elegans in human cells, so these are clearly microRNAs in C. elegans, these primary sequences are making microRNAs in C. elegans. But when we try to overexpress them in human cells, we get a very different result. Many of them, almost no signal for being recognized by the microRNA processing machinery. So it's clear that some of the things that the human cell is looking for when deciding which of these hairpins should be in the microRNA pathway, some of what it's looking for is missing in the C. elegans primicroRNAs. Something different that human cells are using to decide what's a microRNA and what isn't than what the worm cell is using. In other words, what's a microRNA in humans might not be a microRNA in worms and vice versa. So this was a bit of a surprise to us because our computational efforts had actually been pretty successful in finding human microRNAs using an algorithm that had been trained on the primicroRNAs in C. elegans. Okay? But clearly, this program only works when you're looking at conserved microRNAs, so that conservation filter was really very important. And I think part of that is because it is helping make up for the fact that we don't completely understand everything that goes into deciding what is a microRNA in humans. Yeah, or in C. elegans for that matter. And of course, the cell can't decide what's a microRNA based on what's conserved. Okay, if the cell doesn't look at an mRNA, at a hairpin and say, well, this hairpin is conserved to chicken, let's process it. Instead, the cell has to deal with just the primary features of the RNA itself. And so we were interested in this question and basically how the human cell decides what is a microRNA and what isn't. And one thing that we know is very important, and this is shown very nicely by Neri Kim's lab, is that pairing at the base of the hairpin is very important. And what the DRASHA and DGCR8, DGCR8 is actually the same thing as PASHA. It's the human name for the PASHA protein. So the DRASHA-PASHA complex will recognize this hairpin, as well as single-stranded sequence flanking the hairpin, and cleave about 1 helical turn from the base of the hairpin. Okay, and so this is very important. But it actually doesn't help very much in discriminating between different hairpins. Many hairpins have base pairing at their base and single-stranded sequence flanking. Okay, so, but it still is important for determining where the cut is, this measurement of about a turn of a helix. But how does the cell decide which of the many hairpins that can be made are going to go into the microRNA pathway? And so we were very perplexed by this and decided to do what biologists often do if they sort of run up against the wall and haven't really been able to figure something out, and that is to try a genetic approach, okay, to make many different variants and select out those that can still function. Okay? And in this case, our genetic approach was actually done in vitro, where we make many variants in vitro and look for those that could still undergo this reaction. And so we made a large pool of sequences where each position in the flanking sequence here was variant, okay, such that it had a 79% chance of being the starting sequence. We started with the mere 30 prime microRNA. It had 70% chance at each position, 79% chance of being the starting sequence, and then a 21% chance of being any one of the 3 other possibilities. Okay, so if the starting sequence at that position was an A, there'd be a 79% of the molecules would have an A at that position, and then 7% would have a G, 7 a C, 7 a U. Okay, so we had this large pool of variants, but we also wanted the information in the end to be coupled. Okay, so if there was a change at this side, we wanted to know if that correlated with the change at this side. Okay, so the way that we actually did this was to circularly permute this pool so that we would end up with these variants where the information would still be coupled even after the processing happened. So we did this starting with over 10 to the 9 different variants. We made these variants, we incubated them in cell lysate that had the DRASHA and DGCR8, and then selected on a gel those that were smaller and linear compared to the starting sequence, or the non-functional variants here. Okay, so those could easily be separated, and then we could do ligation and sequencing and get the sequences of millions of molecules that had undergone this reaction and compare that to millions of molecules that we had started with, and look for enrichment of some sequences over others. So here, this is just showing you some of the data that came out of this experiment for the mere 30 microRNA, prime microRNA. And you can see that what I'm plotting here is information content, and this is just a function of how enriched the presence of particular nucleotides were in the functional cleaved variants compared to the starting pool. So you can see that at this position at -14, which is just right here, right at the first unpaired nucleotide, there's a strong preference for a U there and a depletion of the other 3 nucleotides, but not much of a preference going further upstream. Checking now on the other end, okay, you can see that somewhere right around here, okay, there's a preference for these 2 C's, okay, C and then 2 residues that where it doesn't really matter, and then another C. So we call this a CNNC motif, N meaning any nucleotide. Okay, and we see this not only for the primary RNA for miR30, but also for miR16. Okay, when we do the same experiment for a different microRNA. And if you know what you're looking for, you can see it here. This is statistically significant for miR223, but seems to be much less important for miR223. And then for the fourth microRNA, prime microRNA that we looked at, we really don't see any preference here. So it looks like different microRNAs are using this motif to different extents. But if you look generally throughout all the human microRNAs, those that are conserved among mammals, we find that of all the dinucleotide motifs that have either 1, 2, or 3, or 0 intervening nucleotides, that this CNNC motif is the one that's most enriched, and it's enriched just at the exact places where we saw that it was important in the miR16 and miR30. Okay, so even though this isn't used for all microRNAs, it seems to be used for many of them in humans. Same thing in Drosophila. Okay, strong enrichment for that same CNNC motif downstream of the microRNA hairpin. Also in Planaria, even stronger enrichment. But not in nematodes, not in C. elegans. So now we're starting to understand why the C. elegans microRNAs were not recognized as primary microRNAs in humans. Okay, and so we've done this type of analysis through, at additional places in the primary microRNA. We find that there's that U that I told you about, and then right next to it, at the first paired position, a G. So there's this UG motif, the CNNC motif, and follow-up experiments have shown that CNNC motif actually binds a specific protein, SRP20, or its relative PRP8. And that binding, we can show, is important for recognition and processing of the prime microRNA. So this turns out to be an SRP20 binding site. We also confirmed the importance of pairing down here, and we did a similar experiment up here and found that for some microRNAs there's this UG-UG type motif, and the pairing is important up here. And so these are the types of things that the microRNA processing machinery is looking for in human cells. So you remember that there are these, and these are not found in the C. elegans microRNAs. And so we decided to take one of these C. elegans microRNAs, miR44 here, and see if we can take that prime microRNA and add in the features that are found in human microRNAs. Okay, so we just we started by sequentially adding in the pairing here that you see at the basal stem, and then the G of the UG, and then the UG, and then finally all four of these, the CNNC, and you can see that the amount of microRNA that we get has increased about 64-fold over what we started with. Okay, so by learning the features that are used in humans to distinguish the prime microRNAs from the other sequences, and then adding those on and modifying the C. elegans microRNA, which is initially not processed very well, we can get it to be recognized much better. So in these types of experiments, we're starting to learn how the human machinery recognizes these hairpins and licenses them for processing. Okay, there's still much to be learned. Okay, when you add up all the information that can be imparted by these motifs and the extent to which they're used, it's clear that there's still more information somewhere in these sequences or structures that we don't yet understand, and so we're still doing these types of experiments to try to figure that out. but at least we have a foothold here. We know that what's important isn't just the base pairing in the secondary structure. There's also primary sequences that are recognized here for many of the human microRNAs. And this is important not just for understanding what's a microRNA, what isn't, for how microRNAs are made, but also for looking at disease association studies. For instance, this C here, this first C of the CNNC in the miR16 primary microRNA turns out to be variant in certain human families that are prone to get a leukemia in the miR16. So having this C different, not a C in the miR16 primary microRNA, seems to make these individuals more prone to getting a leukemia, as shown by Carla Croce's lab, and we now understand why that is. Okay, so thank you for listening to how we are deciding what's a microRNA, and more importantly, how our cells are deciding what are microRNAs. And I just want to then thank these students and postdocs in the lab who over the years have been participating in these microRNA gene identification efforts, and also particularly Vincent here, who did the second story that I talked about, the experiment where he made these many variants and selected out those that could still be processed and learn about these processing determinants for the prime microRNAs. And I want to thank you for listening to this talk, and hope you have a great day.